The gut microbiome, the vast community of bacteria, viruses, and fungi that live inside of us, has become one of the hottest frontiers in medicine in recent years. But while most studies focus on one or a few subjects, this groundbreaking new Cell paper has done the exact opposite. It's one of the largest microbiome studies ever performed, spanning hundreds of species in thousands of human individuals across numerous countries around the globe. What the authors found is both stunning and is likely to inspire years of future research. Global patterns linking the microbiome to how we age, patterns between the microbiome and cancer, and patterns linking the microbiome to the immune system. So what do these global microbiome signatures tell us about the human condition, human health, and aging? Are there truly universal patterns in gut health? And are there regional differences that we need to be paying attention to? Could understanding the human gut microbiome be a missing key to understanding human health, disease, and longevity? If you've ever wondered what secrets the gut microbiome could be holding, this is the interview you should be listening to, because today we are talking to Dr. Sergio Andreu-Sánchez, the lead author on this groundbreaking new paper. Thank you for joining us on the Saving Sapiens podcast. I'm your host, Matt. Let's listen to the episode. And I am so excited today to welcome Dr. Sergio Andreu-Sánchez, the author of a absolutely fascinating new paper on global microbiome diversity. Welcome to the podcast.
Thank you, Matt. I'm happy to be here.
Can you introduce us to the scope and scale of this really epic study that you guys just published, and tell us a little bit about why this paper gives us a much more broad and deep picture of the human microbiome across the world?
I think this goes back a little bit, like for the last 15 years or so, like the field of human gut microbiome has exploded, right? Like we have the technology nowadays to profile what type of microbes live in our intestines. You normally use sequencing technology for this. And for the last 15 years or 2 decades, we've been exploring all the differences in, the composition of microbes that happen in different conditions, right? We have seen that people that have inflammatory bowel disorder have a really distinct population of bacteria, that people who have like mental disorders, they also have different type of bacteria, and that like there's a lot of body of literature that have been relating the bacteria that live within us with all type of our lifestyle, our conditions, etc. I think from the beginning, back in the day, we were only able to resolve back until genus level of resolution. That basically is like if we were saying we're profiling a jungle and we only say how many monkeys do we have in general, right? And after that, we were able to profile a bit more to the species level. So we would be able to say, okay, how many are of those monkeys are chimps and how many are human, for instance, right? But we are overlooking in all these previous studies that we are usually overlooking the diversity that occurs within a single species. So, how many, if we have several different humans in the jungle, which populations of humans are there, right? Or do we have some of them that are diabetic and others are healthy? So this type of resolution is usually lacking in the field, and this is it's a combination both of technical factors and methodological factors that limit us to really go a bit deeper into these genetic differences in the microbes that inhabit the gut microbiome. In this study, what we try to do is to collect a lot of data that has already been generated for the last 20 years and try to go deeper into that data. So sometimes we don't have enough information to go to that depth, but we had sheer numbers, because what we did is to put together around 30,000 metagenomes from over 60 different studies. And in some of the individuals where we don't have enough information to really profile what's the genetics of their microbes. But in general, once you start putting together thousands of samples, you are going to have enough samples to actually be able to profile these genetic differences between them. So we put together all this data set, and then we were able to profile genetic variation in different bacteria, and we did this in over 500 different bacteria. I think previous study that have been looking more into the genetic diversity of the gut microbiome maybe focus on a single bacteria or two. But we tried to do it a bit like large scale here. So we did it with over 500, where we had enough information to have several hundred of samples in which we could confidently like say, oh, this is the genetic makeup of that species. So with this data set that spans different populations, different age groups, different lifestyles, we start then investigating what are the factors that are related with all these what are the human factors that are related with the diversity in the genetic content of the bacteria that live between us. So I think overall, we could say that this is we are revisiting concepts that have been visited for the last 20 years, but in a much more detailed scale that we have been overlooking in the past.
Now, for the majority of the 90s and the early 2000s, a lot of the funding for biomedical research was very concentrated on this idea that we needed to decode the human genome, that we would find most of the answers to most of the illnesses that we were interested in curing by unlocking the DNA in our own cells. Can you give our audience a little bit of a background as a scientist who has a doctorate that actually studied this specific crossover between all these different fields, that we frequently now call this multi-omic studies, where we're going far beyond just the genomic studies? Can you give us a bit of a layman's introduction to why we no longer focus just on the human genome, but why we focus on all of these other metrics, and specifically why the microbiome gets so much attention now? And then we'll dive deeper into the aspects of your paper.
Yeah, that's a really good point. I think it's true that historically our interest was in the genome of humans, and ever since, I think that the Human Genome Project, right, like there was an explosion on research studies that have been profiling, different genetic variants in the human genome and how they relate with different traits of the person or diseases. But what we saw is really early on is that while we identify usually a bunch of genes that are related with different conditions, we could not 100% explain why some people develop the disease or not. So maybe using like genetics, we could explain 20% of the cases. In some diseases, of course, we can explain 100% from your genetics, you are going to have this disease or not. But in most cases, this is much more nuanced, right? We're talking about 20, 30%, or maybe 10%, depending on the condition, like how much we can explain. It's not the same, right? So then it became apparent that there's the genome is not everything. There's a huge component that is the environment where you live, right? Like your lifestyle, what you are eating, like how much sun you're getting. Those things are things that are like really affecting your health and what the type of phenotypes or characteristics that you're gonna have, right? And among them, the gut microbiome appear as a really important one, because we are inhabited by millions of organisms that actually are like everything that we consume, like most of the nutrients are going to reach microbes, and the microbes are going to digest them, and they're going to produce different products that are going to go into our blood and interact with our cells. The microbes themselves are going to be interacting with the immune system. So it became apparent that the part of this, we call it the missing heritability problem, that is like the amount of variability that we cannot explain just from genetics. could be actually attributed to gut microbes. So that's a bit how it came to be that a lot of research from genetics also focus on the microbes that live within us. And of course, the microbes don't tell the whole story either. But I think it also became really interesting for researchers to study microbes, because your genome is really difficult to change, right? So uh, if you have a predisposition for a particular disease, you're probably not gonna go and get your genome edited to avoid that predisposition. But your gut microbiome is something that changed during your life, and that is really easy to actually target, right? So you change your diet, and probably your microbes are gonna change. So I think there was a lot of, and there is a lot of interest in if these microbes are actually related with the development of diseases. We can make interventions that are like really like simple, just some type of diet, some type of product, and therefore target those microbes and like rescue like the this disease phenotype or the possibility of developing disease.
You make such an important point, because the trend of treating diseases as modifiable conditions before genetics was a big focus allowed us to have this idea that medicine could help us escape illness. And with the advent of the human genome research, there came almost a little bit of cynicism, I think, because the excitement was there that we were going to uncover the genetic basis of disease, but then the inevitable cynicism washes over when people realize that you can't edit your genome, at least not yet, not at the sort of safety levels that we would want for humans. And the discussion started to center around almost a genetic determinism or a genetic fatalism, because if you do encounter a disease that has heritable risk factors and they're linked to your genome, we don't yet have a way of necessarily addressing that unless we try to intervene, like with something like a pathological peptide or blocking a particular heritable risk factor. With the microbiome, we have this entire sort of side world of organisms that are interacting with our own gene regulation. And it raises a really interesting question, because one of the mysteries that we've noticed in trends for a long time has been that sometimes people seem to have shared heritability for certain disease patterns, but not based on shared ancestry. And this has been part of the data that has driven people like myself kind of mad when we look at the risk factors for certain diseases like multiple sclerosis or others. There are regional variations in the disease prevalence, even in countries that have mixed ancestry populations, countries like the US, where there isn't a single genetic homogeneous ancestry. And for this reason, this is why when I first read your paper, it leapt off the page for me, because you guys found that there were geographic associations with microbiome patterns. Is that right? Can you expand on that?
Yeah, no, indeed. So, as I introduced at the beginning, we have this really large data set of over 30,000 samples from all over the world, at least in, countries where the metagenomic data was available, right? So we have an overrepresentation of certain type of populations, unfortunately. But with what we had, I think one of the things that strike us the most at first was that when we compared the genetic similarities of bacteria between the participants of the study, what we see is that bacteria that come from participants from similar geographical locations tend to also be more similar between them. So it does seem that individuals that come from the same country also have the bacteria that are more similar between them. And we have a couple of hypotheses why this could be the case. There's one that could be this could just be driven by a common lifestyle, right? So you might have like type of bacteria that are better adapted, for instance, to westernized diet, right? So if you have a westernized diet, you're going to have this type of genetic makeup of your bacteria, because those are the ones that are better suited to consume the type of diet in a Western country. Well, in other places of the world, maybe the diet is a bit different, and other type of bacteria are being selected. So that's one of the hypotheses. But the other, which is I think, at least in my opinion, more interesting, is that these are they are bacteria that have been evolving with us for millions of years, right? So you might have a population that just in the migratory pattern, they brought their own bacteria, and these bacteria stay in that population because they are vertically transmitted or transmitted between closed social networks. So they just remain in that geographical area for a long time, and then they, in that time, of course, they might adapt to specific lifestyles of that population, and then make it like harder to migrate to other populations. And we that's actually we have some data that suggests that this might be the case. What we did is to calculate how often a bacteria is able to go to different individuals. So this is a bit like the an estimation of how easy that bacteria can migrate to other populations. And we see that those bacteria that have more potential for migration between populations are also the ones that show less defined clusters specific of populations. So it seems that when you have like bacteria that have these geographical patterns really well defined, those bacteria are usually not easy to transmit between individuals. So they are probably sticking to a population from the beginning, and probably inherited vertically throughout history.
Wow. Okay. So this is an example. This is essentially a proof case of the. So this is not transmitting across mixed populations in a region. This is more of an argument for a sort of niche co-evolutionary relationship between the host and the microbiome.
Yes, I think that would be my argument. We still need to. So the problem with our study is that when we classify people in countries, is mainly where the data is collected. We didn't have information about their ancestry, because it would be then interesting to see what happened once you go to countries with mixed ancestry. What is actually do you still see this type of regional variation, or then it uniforms everything because the people are in proximity, and then they can still exchange, and you are having a similar diet, so it uniforms everything. And I think that that's a really interesting study that could be a follow-up of this type of findings. But I think there was also a science paper from the group of Ruth Ley in Germany a couple of years ago that showed that the phylogenetic tree of humans, so how similar human populations are between them based on their genetics, like resemble to the phylogenetic tree of certain species. So they mirror each other. So it kind of suggests
It
that this is
literally
happening.
looks like a mirrored tree. It's really fascinating. Very fascinating. Now, the implications that I think got some of the heads turning for health conditions in particular. You guys touched on both things that appear to be associations with living longer and with illness. Can you first tackle what you guys found in evidence of microbiome shifts in the very elderly?
So what we did is using this large scale data, try to find for like patterns in which individuals with similar age groups, if they have similar strains, right? And we found like a statistical evidence for a few species in which we see that individuals of similar age groups tend to have like similar strains. And one of them was really striking, and it was this Ruminococcus navus pattern. And Ruminococcus navus is an interesting guy in the gut microbiome, because I mean, it can be there or not be there, because there's a lot of variability in the population. And if it is there, it doesn't mean anything bad. But it has been shown in the past that, for instance, people with inflammatory bowel disease tend to have like higher abundance of this bacterium. So it's often believed to be a bad guy in your gut, right? Like this Ruminococcus navus. And we saw a really striking pattern in which like the very older individuals tend to have a really, really well-defined like genetic subset or genetic like cluster of these strains. So if you are over 80 years old, it is quite likely that you're going to have this particular strain or subspecies of Ruminococcus navus. And why this is happening, it's not really clear to us yet. We are seeing it there. We see that this is quite reproducible between different populations. So we found it in an Chinese study that they had like really old individuals, and then we look if we can find exactly the same in European populations, and we found the same thing. The only thing is that in these European populations we didn't have that that many old individuals as in the Chinese study, but there's a trend for older individuals in the same cluster or clade in the tree. Um, we don't know if this is related with longevity or this is just the guy that comes there once you are older because your community changes, right? Older individuals tend to have a community that is a bit less diverse, that maybe you are receiving a lot of medications, and maybe this is the guy that is a survivor and it's able to come here and say, okay, I don't care about medications because I'm a bacteria that is really strong to survive everything, and that's why we are finding it there. But it could also be that it's a bacteria that is, because I could assume that maybe older individuals with more than 80 years old that is still taking part of studies, maybe they are the healthy ones. So maybe we have a bias there that we are seeing like a type of bacterium that is more often seen in healthy adult and healthy elderly, and we are picking it up because those are the ones that are participating in the studies. But this is really hard to, of course, come up with an answer for this in a study, a cross-sectional study like us. I think it would be really interesting to follow up people in time, right, for 20, 30 years, and have regular checkups of their microbiome and their health parameters, and see when this transition is happening, and if it is related with a better health or not, or if it is just the bacteria that is able to survive, and that's why we find it there.
Every single time that I discuss or share about a new study, inevitably there's always someone in the crowd shouting correlation versus causation, correlation versus causation. And I think of all of the aspects of this very fascinating, very broad and deep study, I think the desire to have like a demonstration of causation specifically for this finding is going to drive people crazy, because exactly what you said, we don't know if this maybe is the longevity strain that like pushes you to live a couple extra decades, or if this is just like the last survivor, the little guy who just wouldn't get killed off by all the drugs that we're giving the elderly. So man, it will be very interesting to see how the ruminococcus finding plays out.
Definitely. I think it would be really, really interesting to start like even in animal studies to see if we can like give it and see if there's a change in a group that has this strain associated with elderly and another without, and compare if the survival is a bit different between mice or whatnot.
Let's get that study funded. Yes. Well, tell us on the sort of flip side, there's an interest in using microbiome studies as potential early indicators or as potential non-invasive studies for really serious health findings, like in the study of cancer. Can you tell us a little bit about the associations that you found between the gut microbiome and cancer?
Yeah, this is I think a cancer microbiome is a fascinating field. It's also a bit controversial. There have been some issues in the field, unfortunately, in the past, but I think it's also one of the most promising. Like there are I think wild results out there showing that, for instance, you can predict treatment efficiency for against certain cancers just based on the microbiome that you had at baseline. And then there's a whole field out there that they are thinking about modulating your microbiome before you take the treatment to try to modulate the response to cancer. And I think it's fantastic. So in this study, we don't really look much for the outcomes of medication, but what we do see is that individuals that had melanoma showed a really, really distinct a strain or cluster of strains for colon cell lyrofascians. So a bunch of individuals that actually belong to like 3 or 4 different studies that they studied melanoma, and that I think they were from the UK, from the US, from Canada, from Switzerland. All of them, independently of where they are from, they show really likely the same cluster of colon cell lyrofascians once they have melanoma. And I mean, we are not entirely sure about this, but some of these studies, they are treatment naive. So that means that this is not that is being selected by a specific medication in principle, that you could think, okay, yeah, these people having cancer, probably they're having immunotherapy or whatever, and that's why you find the same strain. But some of these studies are treatment naive in principle, so, and we still find the same strain. So I mean, if this is preceding the cancer or not, that would be really interesting, because it could be used as a diagnostic potential in the future. And what is even more well, I don't know if more interesting, but I found fascinating, is that once I was looking into this particular clade of people, like of colon cell lyrofascians that is enriched in people with melanoma, we found that actually people with prostate cancer also tend to cluster in the same clade. So like you find the same strain of colon cell lyrofascians in both people with melanoma and prostate cancer, which we have absolutely no idea why this is happening. Again, we don't know if it's a cause or an effect, but we are seeing this, and I think it would be, again, really interesting to follow up. We did some computational predictions of what type of genes do these bacteria harbor in comparison with other members of colon cell lyrofascians that are not in this clade. And I think that the most interesting finding there was we saw different pathways for vitamin B12 metabolism, but we are not sure, if this is a cause, or again, or just an association. But I've seen in the past some reports that this is also controversial in some studies that found that people with a different type of cancer have higher concentration of vitamin B12 in their blood circulating. So it could be that then this vitamin B12 can reach the colon, and then these bacteria are just able to feed it, and that's why you see them appearing in individuals with these cancers. Or it could be totally unrelated. But I think it's quite an interesting finding that we surely would like to follow up.
There's so many there. This is such a rich and fruitful sort of garden for research questions, both because of the directionality of the causality, if there is any causal driver between, like you were saying, the cancer causing a microbiome shift or the microbiome shift causing the cancer. But even in the years that we await answers to sort of the mechanism and the causal drivers, even if you are able to reliably demonstrate that this association exists, it seems as though this would be a potential diagnostic indicator if that correlation does in fact hold. And I think this is one of the things that I hope to bring more awareness to with studies like this. We often see knee-jerk reactions even from other professionals in the field that we know, well, this is good preliminary work, but until we turn the correlation data into a causal mechanism, we can't really say. And it's not that we're overreaching the data to say this microbe is driving, or this microbiome pattern is driving cancer, or that cancer is driving this microbiome shift. But even insofar as these kinds of correlations that your study opens the door for, even as they become well established, as soon as we have a correlation that is well established, giving that clinical insight to physicians can potentially be a game changer, because man, anything that lets us either track the onset, the progression, or even the remission of any cancer would be data that we would more than welcome in a clinical setting. So I think this is one of my favorite takeaways from the paper, is opening even the opening the field up to the idea that there could be, even in really surprising ways, like the connection between a specific microbiome shift and different kinds of cancers, I think is absolutely fascinating.
Absolutely. I think there's a lot of promising in the field, and of course, mechanistic insights are important for if our goals are to try to treat these diseases, right? To modulate the microbiome to treat disease, but for a goal of diagnostics, a correlation, it's already good enough, right? But then the question is this a cheaper and easier diagnostic material than the commonly used ones? But that's a different question, and I think that there should be other research conducted there to try to answer that.
On the mechanism side, it was also really interesting to read it, because one of the you know one of the most fascinating papers that I remember reading over a decade ago, I want to say it was in 2013 or 2014, specifically talked about the interactions between the gut microbiome via toll-like receptors in the human intestines and the activation of DNA repair enzymes. And the proposition of this paper over a decade ago was specifically that if people were subjected, if in this case, if a study organism like mice were to be subjected to like very strong broad spectrum antibiotics, that the damage and the disruption to the gut microbiome could itself lead to reduced activation of DNA repair enzymes. And we almost have to wonder how many more insights we could gain from finding a mechanism like that. Because if it turns out that I give my patient in the clinic broad spectrum antibiotics and they don't develop a way of reestablishing a healthy gut microbiome, one can only imagine that the role that the microbiome plays on regulating normal DNA repair activity could itself be yet another path to very tragic associations between the gut microbiome and human cancer. I wonder how many sort of future clinical insights we're going to get the more we see the interrelation between these two.
Definitely, I can only agree. Functional work, it's really important. Unfortunately, well, or fortunately, depending how you see it, the work that we did, it's of course really it has no functional readouts, right? We are a bit more like opening door for different hypotheses that we hope that really smart people will pick up to really dig into particular mechanisms with animal models or with cell lines, and really, you know, piece apart what is actually happening there, because we are talking about really complex ecosystems that are interacting with lifestyle, that are interacting with, like all type of variability. So I think it's really important to dig into these fundamental biological processes that are happening there.
Can you wrap us up on kind of the patient perspective? What should everyday regular people know about the gut microbiome and the interface between this very strong push in the 21st century for personalized medicine for the ability of each one of us to kind of have a positive impact on our own gut microbiome health. As a microbiome scientist with expertise in multiomics, can you tell us, are there lifestyle things that we know can help or hurt gut microbiome health? And kind of in the future of things like probiotic supplements and things like that, what kinds of developments should we be looking into if we're trying to best manage our own health and our own health span?
Yeah, that's a great question. I think the field is, of course, maturing a lot. We are now starting to move from the correlation part of things to the causation, but there's a lot of work still to do because it's a really complex community and complex, yeah, like life science is hard. There's no clear, I think, clear answers in the most of the cases. So my general advice is, I think most of this, I would call it classical health advice, it's still true, right? Like eat your veggies. I think it's just becoming more and more apparent how important fibers are for gut microbes and like promoting a good like community that is gonna be good for you in many ways that you cannot even, and we don't really know at this point. But it's becoming more and more apparent how important these like good healthy habits are for not only for us but also for our microbes and keeping that symbiosis healthy. I think it's becoming apparent that early, like these things are important even from, or even more important earlier in life. So if it's, I think there's a huge area of research with infant microbiome and how this might relate with the development of allergies or immune disorders, and how important it is to like try to give like breast milk to your infants, or if that's not possible, then of course I think that there's incredible research on trying to come up with the best formula to mimic maternal milk. So all these things are really important for your microbiome, to reduce the usage of broad spectrum antibiotics. But of course that doesn't mean if you have an infection and you need antibiotics, you need to take them, right? But just like all these things I think are important to have a good healthy microbiome. And regarding products to enhance the health of your gut, I think there are out there a lot of probiotic products. I personally don't know. I mean, they might be effective in particular situations. I don't know if you are like healthy and you have a healthy diet and so on, how much of a benefit they are. But maybe for for people who are taking antibiotics or whatnot, maybe there are certain products that are good to help like promoting the recomposition of the microbiome, or avoiding particular pathogens to engraft in your gut once like if your microbes are you're a bit low in diversity, or maybe it's a bit easier for certain pathogens to come and cause you an infection, but maybe there are certain probiotics that can help in that. I cannot recommend one in particular, unfortunately, but I think that there's a lot of research in that, showing some of them promising results. And then apart from probiotics, I mean, there are more things. There are prebiotics that your fibers and other compounds that they just enhance the growth of the good bacteria in your gut. There are postbiotics that are they are metabolites produced by particular species that you can take, and they are also believed to be good for you. I yeah, I don't have the data in my hand to tell you which one is the one that you should be taking. But I hope that like there will be a good, because I think right now probably all these this type of compounds are not really regulated as drugs are, right? Because it's in principle, you don't. I think the proof that these things work, it's a bit weaker than a drug to be approved and to be commercialized, right? But I hope that in the future we will have a good, you know, process of making sure that all these claims are scientifically sound and that you are actually taking what is best for you. But it's a really complex biology, so yeah, and what is good for one person might not be good for another.
Fascinating future frontiers. Thank you so much for taking the time to join us on the Saving Sapiens podcast and give us a window into this really, really interesting, cutting edge global look at the microbiome and the importance of the microbiome to human health. Thank you.
Yeah, thanks to you.